Hello, good evening, everybody. Good afternoon from wherever you're joining us from across the globe. And welcome to our 15th edition of the Ocean Leadership Series. And today we are going to be talking about something very, very interesting: data management regarding the aquatic environment. And we have just the perfect person to take us through this. Ladies and gentlemen, we bring to you a marine scientist by profession, person of Baki Olalekan Azeez. Please, can we give him a round of applause? Can we give him a round of applause? Can we give him a round of applause as he come on board to take the session? Azeez, you're welcome.
Okay, good day. Thank you for the intro. So without wasting much of our time, I think I'm just going to share my screen so we can start. Okay. Can we all see my screen?
Yes, we can see.
Okay. Thank you.
Yes, sir.
Okay, so good day, everyone. Today we will be diving into the data management in aquatic environment. So I'm just gonna. Okay, so to begin with, we need to understand the aquatic environment. So we know marine aquatic environment simply means the body of water, and then we know that the earth is being covered by approximately 75% by water, and we have different categories. We have the fresh water, we have the marine water, and the other small water body. And then this environment is a very important environment because of several reasons. First, they serve as hotspots of biodiversity. That is, there are varieties of living things in the environment, and then they also part of CO2 sequestration. As we know, CO2 is a chief source of climate change, and so our aquatic environment also absorb this excess CO2 in the atmosphere, and also as a source of oxygen to the atmosphere through the activities of the phytoplankton and other green plant species. So we also have the function as a source of energy through the wind farm. And we also have other activities such as the fisheries, aquaculture, as a tourism, as also shipping for the transfer of goods from one part of the continent to another. So we see that the environment, the aquatic environment, is a very important environment, and that leads to the reason why we need to conserve and manage this environment for us to have the continuous service provided by this environment. And so for us to be able to conserve and manage this environment, we need to have the ideal of what is going on in the environment. And how do we get to the idea? We need to have knowledge about it. And of course, we can't have knowledge without being informed. So we need to inform. And what can give us this information is data. So then when we say data, data simply means a raw fact about a phenomenon or about something. And in the context of aquatic environment, we can have different data that may be the biomass, the soft oxygen, the temperature, among other things. And this, we cannot have actual information or idea of what this is all about. It is just like being giving you this data and then you don't know what to bring out of it. So this is just data and they are raw facts. And then when we come together and we process this, we get information from this. For instance, we can see those in yellow maybe discussing or maybe describing the physical properties of the environment, while those in green may be describing the biological component. So from here now, we have the information of what is each of the data is talking about. And when we go further, by connecting all this together, we can have the knowledge of what is going on in the environment. And finally, we can have the factors. For instance, when we are looking about the influence of these parameters on biomass. So we may be determined that, oh, maybe dissolved oxygen is having impact. So from this, we have the idea. And if we want to conserve a particular thing in the marine environment or in the aquatic environment, we know where we can focus our energy to because we have the idea of what is going on in the environment. So that leads us to another section where we talked about where the data comes from. We talked about data, but where can we get this data? Are they just anywhere, or how can we get it so we can get ideal and make decision from it? So in aquatic environment, there are different ways in which data can be provided or where you can get data. For instance, you can get from data from satellite. There are different satellites that is being owned by government body, and then they have a server where you can download the data from the data you need. For instance, the Copernicus data, so you can get the data on the particular environment, specifically in the marine environment. So we can also use drone to also get data. You can use drone to monitor the marine or the aquatic environment, and you get the particular data you want. We can also use probe, such as we use it to measure the parameters like the temperature, the dissolved oxygen, among other parameters. We can also use CTD sensor. So this is just to measure the depths and the other physical parameters such as the temperature, and you can also use it to collect the water from the bottom. Then we also have the Nepskin bottle. This bottle is just used to collect water for maybe phytoplankton or for other parameters you might be interested that is in the water body. You can also have the Van Vliet graph. So this is specifically for sediments. If you are looking into maybe the benthos, the species that are found in the benthic in the sediment, you can use this to take the sample, then you can analyze and get the data from it. Then we can also have data from the autonomous underwater vehicle. So this is just going to be deep into the depth of the water, then start to record the parameters that has been calibrated for it. Then we can also secure data from the monitoring activities. It may be annual monitoring of a particular water body, or monthly monitoring, or weekly. So from here, you get to know about the different species, you get to know about the condition, about the different activities. So from there, you can also secure data. Then we can also secure data from the laboratory measurements, where you just take the water sample to the laboratory and you measure different parameters such as the PFAS and other POP. Also from tagging, we know that there are some research that that is dealing with tagging of fish, for such as the migratory fish and other living other aquatic organism. So they are just to monitor it, and then to get, you can also secure data, and the data can also be used for decision making purpose. And also some of the advanced method of data is just the the DNA. It may be in form of the environmental DNA, which is known as the eDNA. And then this is just to get the footprint of the environment. It may be in form of collecting the DNA of the organism or the DNA of the environment. So from here, you can determine what is going on, like in terms of evolution, in terms of the ecology and the rest. Yeah, you can also have in terms of citizen science. So this is what we can say we are concerned about. That is, you don't need to have all these gadgets or all this equipment before you take data. You can just go to the environment or to the water body, and at least perhaps you can ask people around some questions, or you yourself, you can just devise, okay, I want to measure this, I want to take these parameters, I need this information. So can also be a source of data. And then finally, we can also have the planting net. So this is just for the planting, like zooplankton and phytoplankton. And then this organism or this group of living things, they are very small, and they can only be observed with the use of microscope. So from there, you can know the functionality of the environment, of the aquatic environment, and how they are being affected, because this group of organisms serve as a bottom for the food web. So, and then, so I haven't know about the different ways we can get data. The next thing would be, what are the data we actually need? Because we know that we can get different data based on the different equipment and based on that. But the data we actually need will depends on the question we are being asked, or the question we ask. So with this led to the Copernicus. According to Copernicus, they divide the environment into three. We have the blue ocean, which is describing the physical state of the ocean. And for this part, you can talk about the salinity. You can be getting temperature data, the wave, the wind, the sea surface height, velocity, among others. So when you have this type of data, it simply means you are describing the physical nature of the environment, of the water aquatic environment. You want to know in terms of physical activities, in terms of what is going on in terms of the physical activities for you to make a recommendation or for you to make a decision. So then you can be talking about the biological parts, which they classify as the green ocean. And in this, you may be looking at the chlorophyll A. You may be looking at the light attenuation, how it affects the living things, mobility, zooplankton, nutrients, the source oxygen. You may be looking at how these parameters is affecting the living organism in the environment. And then the last part is talking about the white ocean, which is those in the polar region. As we know, the polar region serve as a shield for excess heat. So you may be interested in determining the concentration of the sea ice or the velocity or the albedo, the reflection of the heat, and then other maybe the thickness. So all these are the classification given by the Copernicus in terms of the data you can have in the marine environment. And then, so most of the time, the data we always have, they are mostly unstructured data and they are very large, maybe greater, more than 20,000 rows. And you might also be having to combine different data sources. For instance, now the data in the left side of the slide, you can see that you have different stations, and you have the different species that is being taken from different stations, as well as the date and other information. But this data, you may look at it and think it is structured data, but it is not yet clean. There are still some error. You will see that we have the density biomass, we have the value, you have the unit, but you will need, as you can see, the tab under here in which the biomass and the density are being separated. So this is one of the ways in which you get your data. You have a large amount of data that you need to clean, you need to remove, you need to do a lot of things, which we are still going to talk about. And then most of this data comes in different formats. We can have it in form of Excel, or in form of notepad, in form of CSV, or even in form of the geographical data like the spatial data. Also, it can come in form of PDF. So all this might be, you may have data from different source and you need to combine them together for you to be able to use. So this led to the management of the data, that is the challenges that we can face when we have different data that we want to look into and we ought to have idea about. So some of the challenges might include, what data do I need? Where do you get the data from? It may be that, okay, you get one data from this source, you have to look for another source to get another data to complement the one you have. And then which format is the data? Do you have to convert all your data to to Excel file, or you have to convert it to CSV or other format that might be of interest? And then the unit of measurement, you need to know the units you're working with. You may be giving data, for instance, the temperature, you may be measuring in form of degrees Celsius, and another data may be given to you in form of degree Fahrenheit. So you need to convert, you need to work with that. Another challenge may be, maybe you have different names or the same name for different roles. So you need to look at, that's one of the challenges. Also, when you talked about the missing data, there are a lot of times you will have missing data in your data set. So you still need some of the challenges that we encountered in management. So, but how do we go about it? So I'm just going to highlight some of the step-by-step in which we can manage the data and in which you can get the best of the data, and then we can use it for onward analysis and decision making. So the first is you need to understand your data, then data structure, then data standardization, and data validation and analysis, and at the end, the report. So let's start with understanding our data. So as I've said before, we first of all need to understand the data we need. What are the parameters we want to measure? For instance, if you are interested in the physical chemical nature, you might be interested in having temperature, in having turbidity, salinity, and all this is just talking about the physical nature of the aquatic environment that you want to study or you want to have idea about. Then you can be informed of the biological variables. Maybe you want to look at the particular influence on the fish, or maybe in terms of the benthos, or in terms of zooplankton, or in terms of phytoplankton. So these are the data that you might be need. This is not the only data. There are a lot of data that you can also have in terms of biological variables, but this is just part of it. Then you can also have other data like the attenuation, like the POP, or maybe you're looking at the plastic. You may collect data in terms of plastic. It may not necessarily be in terms of going to the aquatic environment and taking plastic. It might involve questioning people about their knowledge. It might involve interacting with experts. So those are also source of data and the data you might use. So, and then how do you structure your data? Often the data is always structured in this format, in which the column will be your variables, that is the parameters you have, while the rows will be your observation, that is the different observation for each particular variables that you mentioned. And example is this: when you have the time in column A, and these are the time in which these parameters, the pressure, oxygen concentration, oxygen saturation, conductivity, and the rest, they are measured. So you can see that at each particular time, they are all having the same, they are all having particular value. So this now you can go ahead to have other look, since you understand your data. Okay, these are the parameters that I need, and this is talking about the physical parameters of the aquatic environment. So the next is to standardize. Now, when you are trying to standardize your data, as I've said, you will have data that might be unstructured or dirty data, we say, or data that you have different names in the data, but you need to homogenize your data in order for you to understand and to be able to use it for onward. And also, it can also be shared among other people that can also use it. So the first thing is to set a standard for points or station nomenclature. That is, you have to have different stations that you are measuring. You may use point one, point two, three, and the rest, or you might use S1, S2 that to stand for stations. If you have several stations, you may just use S to represent them, or perhaps if you are measuring from different river, you can have river one, you have 1 point in river one, you have river one again, you have another point, another in same river, you are mentioning another point. So like this, anyone taking the data will have the idea that, oh, I think this is about river one and this is the point. So it can easily be read by any person taking the data, and for you as well, for your onward research or for onward process, it will easily facilitate working with the data. Then the next thing is to establish list of variables. For instance, it might be very not easy to write all the variables name. You can just shorten it by just using temp for temperature, or DO for dissolved oxygen, or DOS for dissolved oxygen saturation, or night. Just use something that you can easily understand, or you can easily remember what it stands for. So then the next is to set the unit. As I've said, you might have different unit of measurement, but you need to have a particular one that you can use. And then, for instance, if you get a data of the source oxygen, you might get one that is being measured in milligram per liter. You might get another one being measured in liter. So if you are not having same unit, you might go ahead to analyze and give different results, which may lead to wrong decision making. So you need to first homogenize your units to check if they are all the same unit for onward analysis and decision making. So then I talked about missing data. So mostly you always have missing data, not for different reason, but how do you deal with missing data? This is just a screenshot of how to check, because if you have like thousands of rows, for you to be scrolling down, that'd be difficult. You might just use the filter format in the Excel. So just clicking on this filter formats, you can see blank or NA data. So then you can easily go to that point to check what's going on there. If it is missing data, if you are going to need to remove it, or you have the idea of what should be there. And there are several ways in which we deal with missing data. You might, for instance, let's say this gravel, there is no data in the row 4. Let's say there is a missing data here. The ways in which I can do it is to find the mean of the row of the column and insert the mean there, or perhaps I can just remove it, or I use other advanced method of handling missing data. So, but I'm just showing the ways in which you can quickly search for missing data in your data set. So the next thing is also to take attention into the date format. Different region have different ways of writing date format. It might be in some region, maybe you have a data in which the month comes first, followed by the day, or you have the day first, followed by the month. So you need to also look through this and see if the data or if the date is well formatted. So then we talked about the duplicate. Yeah, so this is also another challenge that we need to look into. So how do we do duplicate? There are different ways you can handle duplicate, but I'm just showing the in the Excel where you can easily identify your duplicate is just by coming to the table, and then you can see to the data, then you can see this icon. So clicking on this icon will highlight the duplicate and even remove it for you. So with that, you are cleaning your data and you don't have repeated data or repeated rows. So then the next thing is saving your data. How do you save your data? After you clean your data and do necessary things, and you are certain or you are sure about the data that okay, to your knowledge, this data is okay for onward management. You can save your data and you can easily be able to locate it and then be able to know where you are with the data. For instance, now if I have data from Onion River for River 1 and I collected the data in 2010, I can easily name it here. So anyone taking or checking my folder to see the data, we know that, oh, this is a river from Onion, or this is Onion River, and this is the first river that I'm talking about, and this is the year in which the data is being collected. Or perhaps I can use clean data underscore the date of the date in which I clean the data. So anyone I'm sending the data to will know that, oh, this data has already been cleaned. And then, or you can use the name of the project you are working on. Maybe you are working on invasive species and you've cleaned the data, or maybe you are working on plastic and you've cleaned the data. So you can easily name it. So you'll be able to differentiate it from the other data that you have not cleaned. And when you get new data, you'll be able to know which data is the one that has been cleaned and which one has not been cleaned. And then, so the next step is the data validation and analysis. And for this just to check if your data has already been standardized, you have done all the necessary things to make sure that they are all in good shape. And then also duplicate, you've removed the duplicate, and then you yourself, you know that, okay, this data makes sense and well structured. There is no any missing data. You've figured that out, and then you can go ahead. So this is the first thing we do when you have data. And then this is called the quality control and assessment. That is, you check the data for any error, for name adjustments, for duplicate, for standardization, for unit, and then other things before you go ahead. Because once the data is being affected or it is not well structured without quality control, then that might lead to an error, or that may lead to wrong decision making. Because imagine now you analyze the data that is not clean and you came up and tell us, oh, there is no plastic in this water body. Meanwhile, there is plastic affecting this organism. And another person came up and say, oh, and then from your decision, the policy or the decision maker has taken note that, oh, this water body is clean, so there is no any error. You people, you can go and take the water, you can do anything. But you've already given wrong information and the decision has been wrong, decision has been made. So that is why you first need to check the quality control of your data before you go ahead with other step. So now the next step is to conduct analysis and visualization. And then for the analysis, there are different analyses, but we are not going to be going deep. We are just going to be talking about the different analysis you can have. We can use regression. So regression is just to check the influence of one variable on the other to have the idea of what is going. For instance, now in this case, you can see we are talking about the influence of years on the number of non-indigenous species, and we can see that as the years increases, the number of non-indigenous species is also increasing. So we can say in this particular water body, the number of non-indigenous species will keep on increasing as the year increases. So there should be a management from here now. So the next thing is also correlation. For the correlation, you may be looking at, okay, is there a relationship between different parameters you measured? Okay, maybe you're talking about the temperature and the dissolved oxygen, or you are talking about the dissolved oxygen and their salinity. So you want to know, is there any relationship among them? So you might do something like the heat map to show the correlation when you have the one greater than 0.5 or below 0. I hope I'm communicating.
Yes, you are.
Yes.
Okay, thank you. So then another way in which you can show your data is using bar plots. Here we are showing two things on one plot. This is another way of managing your plot. Instead of showing it separately, you can show the two together. For instance, now we are comparing the amount of native species and the amount of non-native species. So, and we can see that as the year increases, the number of non-native species, which are in red, is also increasing. They are becoming obvious. So this is another way for us to bring about the decision that, oh, this is also validating that as the year increases, the number of non-indigenous species also increase. But for native species, they are kind of constant. And you can even see from here that it is low in this particular year. So from here, anyone you are presenting this to, we know that, oh, this is what is going on. And then proper decision can be taken. Another way is it can also be not only in form of a graph, it can also be in form of spatial data analysis, in which you have the spatial layer. It can come in form of you recording the coordinates of the particular point, or downloading the shapefile from online and just using your own data, putting there. So it will give it in form of the like your Google map. So you have your Google map, and then you can tell, oh, at this particular point, there is a lot of traffic, or at this particular water body, there are a lot of fish, and then different things you can do. So it's aids visualization and it's aids understanding. So for instance, now this chart here is talking about the different estuary water body, and then the number of species that is being gotten from each particular point that is being sampled. So from here now, you can see that those points that are bigger circle and those points that are smaller circle. So then another way is to just show something like a circular plot. So then you show the number, and then the bigger the number, the bigger the circle. The smaller the number, the smaller the circle. So you can see from here that this particular species is most abundant in this particular water body, or this particular species is less abundant. So this is just the visualization. Then there are also ways in which you can show your data without showing plots. It may be in form of table. For instance, if you are using hypothesis testing, such as the ANOVA, the parametrics, chi-square, among other. Then you can also have multivariate analysis when you're talking about the community analysis, when you're talking about the ECA and the rest. So, and then you can also use machine learning. Yeah, machine learning is becoming more and more used in the analysis because of the being robust. You can use maybe random forest for classification or for regression. You can use among others, but we are not going to be going deep into this analysis, but this is just to have the idea about how we manage data in the aquatic environment and how we can present it to the audience or to the decision maker. So then all these analysis can be done using different software. You can use the RStudio. You can use QGIS when you're talking about the geographical data. You can use Excel. Yeah, Excel is a very important and very robust tool that you can use for your analysis. But RStudio is for programming. If you are good in programming, you can use RStudio, or you can use Python. So, and then you can also use another one, JAPS. So this one is just for you don't need to write any code. All you just need is just to click and drop, then you get your results. And then you can also use Primer 7. So these ones are used for community analysis if you are into ecology. So you might be interested in using this Primer 7 for easy analysis of your data. I will be stopping here for today. So the next step would be to, after getting your data, is to write your research or to write your report about what you find out. And then you have to recommend what should be done for this particular situation or this particular thing you research on. But so that's not the focus of today, but today is just to have the idea or the knowledge about the management of aquatic data. So I would like to appreciate the Water Service and Technology for the idea about the management, and also the Copernicus Marine Service for using their data, as well as the Aquaworld and the ASTI, where I did my work. So to everyone, I say thank you, and that will be the end of the presentation today. So if you have any question, so the floor is open. Do we have any question?
Yes, sir, I have a question.
Okay.
Can I go ahead?
Yeah, you can go ahead, sure.
My question is that you talk about machine learning or Python when presenting the data. So is there any specific app that we can use for the, like, is an AI that you just input your data, it will do everything for you, or you have to work on that for yourself? That's my question.
Okay, so as you know, this is an era of AI. So definitely there is AI you can use that will give you all this, but I will advise to use it with caution, because AI is just a programming language, or it's just artificial intelligence. You need to have the human idea of what is going on. For instance, now, if you have a data that you need to standardize, maybe you have 100 data, 100 here, and you have other point that is 2, 3, the 100 is going to have impact on your analysis. So you need to standardize that point. But once you give it to AI is not going to standardize that unless you tell AI what to do. But as a human knowledge, you will know, looking at the data, doing the quality analysis, you will be able to see that, oh, you need to standardize this point, you need to remove this, you need to convert this from one unit to another. But once you just give the data to the AI, it's just going to do the analysis without checking the data if it needs to be formatted, if it needs to be standardized or any other thing. So yes, there are apps, there are AI you can use, but you need to use it with caution.
Thank you so much. Thank you.
Okay, a very enlightening session, sir. We were able to access the slide. Okay, so that has been answered. So is cleaning data and standardization data a different activity? Yes, they are different, but they seems to be close. Because when you talk about cleaning data, it means you are removing some redundant data, some data that you think is not necessary or is not relevant to your data, or you are removing missing data. But when you standardize, you might be standardizing in terms of, maybe you look at your data. For instance, now you know that your temperature of a particular water body should be within this particular range. And when you check to your data, you found a range that is very high than the other one, which might serve as outlier. So it's either you remove it or you check if it's actually what it is. So, or it may be like you have a data and then you see that, for instance, if you are taking the species data and you measure the biomass of those species, and you find out that one of the species, okay, you have two species that they are not the same weight. So one may be weighing 100 gram, while the other one may be weighing 2 gram. So you will know definitely, if you are conducting your data, the one with 100 gram will have impact on your results. So it might require you to standardize, and there are different ways in which you standardize your data. You can use square root standardization, or you can use log standardization. So with that, you'll be able to have a balance or weighted data that in which one is not affecting the other one. So that is about standardization of your data. Okay. Yeah, you're welcome. So do we have more question? You can drop it in the chat box as well. Yes, log and square root. Okay, thank you. So if we still have question, you can unmute yourself and ask, or you drop it in the chat box. Thank you. Yeah. Yeah, it's very sure because it's just to have the idea. We don't plan to go deep. So if we are to go deep, it's going to take much time. So, but this is just to have the idea about how you can manage your data, how you can collect the data, how you can come up with the idea of, okay, you notice something in the marine environment or in the aquatic environment generally. So what can you do to assist? So it's not necessarily you have to have a particular project in mind. You might just decided, okay, we all know that there's climate change, there's climate change. So you want to just have a record of the data. That is one of the things that we need to take into account. It's not necessarily until we have a problem or we have issue or challenge to solve that we need to take data. We need to have a record. I've seen a research that they use a record of 1985. Imagine collecting data at that period. So, and most of the data comes from citizen science. So it's just people noticing something in their environment. Oh, we found this fish, which is unusual. So they note it down. So in the next time, perhaps who knows when you're going to need the data, then you can consult them and then get the data. Oh, we've noticed this species since this particular period, and since then it has been occurring. So that is also the way you get data, or you note some things. Okay, go to a particular water body, you observe, oh, this water is at this level, this period. Then next time you measure or you look at it, you find another level. So at the time, there may be a research in that particular water body, and you can also provide your data. Oh, this is the data I've collected, or this is the data I observed or I taken. Then, so it might also be used. Community analysis. Yeah, community analysis or community ecology is an analysis that is done for the ecology of organisms. So community simply means different population. Am I correct? Or different organisms coming together. Then you look at, oh, which of these organisms is abundant, which among it is diverse, and then yeah, if you're talking about diversity, there are different indices you use to measure diversity. And also you may be looking at their interaction or the influence of environmental parameters on these different species or different organisms you have in that. So that is just like a basis of community analysis dimension. Yeah. Okay. So what's time? So it's already I think 5 p.m. in Nigeria time. You know? But if you still have question, you can drop it in the text box, or you can unmute yourself and ask. So I think there is another question. Lovely presentation. Thank you. Thank you, Salvador. So, thank you also for attending. Thank you. Okay, so I guess we don't have much question again. So I would like to end the presentation now. And then if, okay, do we have any question? It was enlightening. Thank you. Thank you for that. So I don't know if the Aquaworld chairman will want to say something. Well, okay. So I think this will be the end of the presentation today. Thank you for everyone that took their time to join the presentation today. So we hope to see you next month for another beautiful session. So thank you. See you all.
Thank you.